So as we begin to monkey around with DNA, I'm always amazed at when I think of our genome and our genetic code. Our genome are all of our genetic material in all of our cells. And so to put this into a little bit of perspective, when we look at a single human chromosome, these are linear DNA molecules. If you could stretch the DNA all the way out, one human chromosome would be 5 centimeters long. If you then think we have 23 different chromosomes in our body, 3 different types, we number them 1 through 23. If you were to put them end to end, now we're up to 11.5 meters. But remember, you got 23 chromosomes from mom and you got 23 from dad. So you have 2 copies of each, so you have to double that to 23 meters. And then when you think of 10 trillion cells in your body that has 23 meters of DNA, you link those end to end, now we're up to almost 1.5 billion miles of DNA. To go to the moon, you're going to travel 250,000. The sun is 93 million miles away. So if you took all the DNA and all your cells, put them end to end to end, it would reach to the sun and back 7.5 times. That blows my mind. That's a lot of stuff. And remember, everything that you are and everything that you do is wrapped up in all of those billions of miles of DNA. So the question is, how do we get 23 meters of stuff into a little bitty teeny tiny cell? And that's packaging. And we're going to start the packaging of our linear DNA molecule by wrapping it around little proteins that are going to act like spools. Have you ever seen the old wooden or plastic spools that thread comes on? Well, I want you to think of these proteins that are called histone proteins, much like you would that wooden or plastic spool. And instead of thread wrapped around, we're going to wrap DNA around it. And we're going to wrap, maybe about 50 or so base pairs of DNA around these proteins, and it's going to look like beads on a string. So with our histone proteins and the DNA that is associated with those proteins, we call that bead. That is called a nucleosome. So do you see already how we're shortening our DNA strand? Just wrapping it around these proteins, creating the nucleosome. That's going to shorten the DNA strand. That's a first level. And so this just gives you a little bit of an idea. Here we have our histone proteins, and there are going to be about 8 of them working together to create the spool. We're going to wrap some DNA around it. Then we're going to have a stretch of DNA that's extending out. And then we're going to have another bead, and then we're going to have another bead. And this goes on and on and on and on as a first level of packaging our DNA. But that's not the only level. Because we start with our nucleosome that we just described, our histone proteins, the DNA that it's attached to. And then we're going to take this string of pearls and we're going to create a spiraling fiber. And so we really don't see the space between the histones here. That space is just enough to wrap that fiber into a tight little bundle. Again, shortening the DNA. But then as we pull back our magnification more and more, we're going to take this fiber that's created and we're going to begin to loop it. And as we loop this around and around, we're going to loop it and bend it and twist it until we end up with something, when you look in a microscope, it's this dark staining material that you see in an electron micrographic picture that we call heterochromatin. But you see how we're just condensing it further and further and further, compacting it further and further, until finally we have the most highly condensed state we can have. And this is going to be in the middle of our cell division, where we have 2 copies of a chromosome still attached together. Many textbooks refer to this as a chromosome. We're going to modify that terminology a little bit when we get to our cell division chapter next week. But just as the cell's dividing, that is going to be the point at which your DNA is as highly condensed as it can possibly be. And that's how it fits. 23 or so meters of DNA fit inside every nucleus you have in every one of your cells. And it has to be there, or else that cell would not be living, would not be able to function. To me, that's pretty amazing. Also, that number, 10 trillion cells that make up your body. I can't even think of that many zeros behind that number. And it all works together, and it's all balanced, because what is that balancing word that we're all trying to attain? Homeostasis. 10 trillion. Sometimes we can't get 3 people to agree. Imagining getting 10 trillion cells to all work together. A very, very difficult task indeed. But it happens each and every day, every minute, every hour, your entire life. All right, so that's our little synopsis of DNA. Now it's time to make a copy. Now we're on the path of making proteins. And to make proteins, we have 2 steps. The first step that happens in the nucleus is transcription. Transcription is making a copy of DNA to RNA. Why do we have to make a copy of our genetic code? So, whenever we're making a copy of the genetic code and we split into daughter cells, we've got to make sure it works. We have to make sure it's good. But in this case, why are we making a copy as RNA? Why don't we just make another DNA copy? Okay, RNA transfers from where to where? Yeah, we've got to get out to the cytoplasm, because that's where our protein factories are, right? Who are our protein factories? Ribosomes. And they're in the cytoplasm. So we have to copy that code so that we can ship it out to the cytoplasm. And that code's only going to be for one specific gene, one specific protein. We're not making codes for a whole bunch. It's just the one we need to make. And when that gets shipped out to the cytoplasm, that's where we're going to translate the code, translate it from nucleotide language into an amino acid language that allows us to make a protein. Now, we've got a couple of different ways to look at this. Whoops, didn't mean to go that far. Here we see our cell. This is what we started with, the very beginning of our chapter 4. We can see our nucleus. And within the nucleus, our double-stranded DNA. And this is sort of a static process by which we use an enzyme to break the hydrogen bonds and separate the 2 strands of DNA. Use one of those strands as a template upon which to build a copy of our RNA. Then we ship that RNA out of the nucleus and into the cytoplasm, where we work along with ribosomes and another partner that we're going to talk about, transfer RNA, to actually assemble the amino acids in the right sequence. So protein synthesis relies on these 2 mechanisms. Without either one, you would not get a protein. You've seen these terms now 3 slides in a row. What do you think that means? It's important, and you're probably going to see it again where? On the test. Transcription in the nucleus, DNA to RNA. Translation in the cytoplasm, RNA into proteins. Those happen within the ribosomes. Now, as we do this process, and transcription we're going to cover first. It's first in the sequence, but it's also less complex than translation is going to be. We're going to have a number of steps that we go through. And as we look at making this RNA copy, it's important to understand DNA, those 23 meters, it would be crazy to try to transport that whole complex to the cytoplasm just to make one gene. If you go to the library and you need to copy one page from a book, you're not going to carry this whole big huge book out with you if you just need that one page of information. You're going to walk in, go to that page and do what? Make a copy. Snap a picture with your phone, walk out. Well, our mRNA, our messenger RNA, the copy of the code, is exactly what we're talking about. It's a copy of one page, one gene for one protein. And it's particularly useful because that one copy is small enough to get through our nuclear pore. And so when we look at our DNA and where we need to copy the code, there is going to be a place where the enzyme that's making the code is going to know, I need to start here making the copy. And there's going to be a part that says, okay, I'm done. That's all the information I need. The starting point's called a promoter. It's like the landing pad for the enzyme. It's not actually part of the gene, it's in front of the gene. It allows the enzyme to sit, move forward a little bit, and go, okay, now I'm ready to make this copy of the gene that's going to be useful for making a protein. It does that until it reaches a stopping point, and at the stopping point, it knows it's copied every single nucleotide needed that's going to be used in the ribosome to assemble a fully functional protein. Because if you're one nucleotide short, that could mess the whole thing up. It has to be intact, it has to be complete. And so this promoter start and this terminator stop for transcription are as important as the gene itself. All right. Now, before we get into the slides and the stepwise process, I found these really nice computer animations of the processes of transcription and translation. So let's watch them first. They're just little short clips, a couple of minutes. And then we'll go break down the steps and talk about the specific components that are making this happen.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm.
Transcription. Did you see in the assembly process how this enzyme took nucleotides, the same alphabet, essentially, as we have for DNA, and made a copy of the code. And that's essentially transcription. And so, in a nutshell, we are looking at our double-stranded DNA containing the code is going to be used as a template that we're going to copy from. We have a sense strand and an anti-sense strand. Remember, they're running in opposite directions. The sense strand is actually going to be the exact same code as we'll see in the messenger RNA. So that means to assemble the RNA, which can only go in which direction? Which direction can RNA and DNA only be synthesized in? 5' to 3'. And that means the template's going to have to be the antisense strand. And that's what it's there for. To be the template upon which we build our RNA copy. Now, we said it's the same alphabet, but there's one important difference, right? What's the difference in RNA? DNA has a T. RNA has a U. That's our only difference. So in RNA, we look at A and U and C and G. But there's also another difference in our RNA molecule versus DNA. Our DNA is a double helix, right? 2 strands. How many strands does our RNA possess? 1. It's only a single strand. So these are important differences. You use ribose sugar instead of deoxyribose. You have a uracil present instead of a thymine. And RNA is single-stranded versus double-stranded. Now, in this process of separating the DNA, breaking those hydrogen bonds, assembling the nucleotides into our RNA strand, this is a fairly simple process because it's all done by one enzyme called RNA polymerase. Because remember, our nucleotides are our monomers. Our RNA strand and our DNA strand are the polymers. Hence the name RNA polymerase. So the name is pretty straightforward. And really the process is pretty straightforward as it's happening in the nucleus. But I said this is an enzyme, RNA polymerase. What category of macromolecule are enzymes? Proteins. Where are proteins made in the cell? They're in the ribosomes. And where do the ribosomes live? Cytoplasm. So if we have to use an enzyme to make an RNA copy, and that enzyme's a protein, you see it's going to have to be made in the cytoplasm, and then somehow that protein is going to have to be imported back into the nucleus. That's going to be those nuclear pore complexes and that sort of allowance to get RNA polymerase back inside. So let's break this process down that we saw in our animation. We're going to break transcription down into 3 very simple steps. And the 3 steps that we're going to have for transcription are initiation, elongation, and termination. And we're going to take these one step at a time. We're going to add some details to each one of these as we walk through our 3 steps of transcription. So let's start with initiation. So for initiation, RNA polymerase has to bind. And where does RNA polymerase know to bind on the DNA strand? The promoter site. Now, I'm throwing in a term here that you may not be familiar with, but this term is called upstream. Since we have a reading frame of our DNA, 5' to 3', you can almost think of that as water flowing in a stream. And so if our gene is at this point on our DNA strand, anything that's going to be 5' upstream, do you see what I'm talking about? the beginning, before you actually get to the gene. That's where the promoter is. That's the landing site for our RNA polymerase. And when RNA polymerase binds to the promoter site, it's going to begin to break the hydrogen bonds between the two chains and separate the double-stranded DNA. So now we can use the antisense strand to make a copy. We're going to use that upon which to assemble the nucleotides. And the assembly of the nucleotides is step 2, elongation. So as RNA polymerase is riding along this rail that is our strand of DNA, it sees, okay, the template's got a C. What is RNA polymerase going to put into its RNA strand? If there's a C on the template, it's going to use a what? A G. All right, let's walk forward. Here's a T. This is a tricky one. Here's a T on the DNA polymerase. What are we going to put in our RNA? A. Because you have A in your RNA. So if you read a T on the DNA, you're going to put an A in your RNA. Now, here's the one that's the RNA trick. The DNA has an A in it. Are we going to be able to put a T in our RNA? No, what's going to go there? The U. Read again, there's a G. What do we put? C, and on and on and on. That's the elongation. Matching them up, our DNA for our RNA over the course of the entire gene. And that's going to continue until we get to step 3. And essentially, what is step 3? The stop, the termination. It's like RNA polymerase is going along, and it gets to the point of the gene where there's an oil slick. You can almost think of it like that. It gets to this slippery space that's got this sequence that says you're done, stop. And when it gets to that place, it just lets go. And when the RNA polymerase lets go of the DNA just snaps back together double-stranded, and we have a free-swimming, free-floating message code copied into our RNA. And this particular RNA is called messenger RNA, or mRNA. The RNA copy of the code is mRNA. Now, did you notice toward the end of our video, the video showed pieces of the RNA being clipped out, and then the pieces that were left pushed together? Well, this is something that happens after transcription, and it's going to be the clipping out of segments that are called introns. Introns are junk pieces of DNA, or in this case RNA that we've copied, that are in the way. That's the way I link intron, in the way. We've got to get them out of the way. The exons are the exciting stuff, the good stuff that's going to be used to build the gene. And so this process of removing our introns and splicing the exons back together is called just that. It's called splicing. Does that sound like a lot of work? A lot of trouble? Why even have these junk pieces of DNA around? It's a lot of trouble. You could make a mistake cutting them out. What if you cut in the wrong place? We've already talked about cancer before. What causes cancer? Destruction of the DNA. There's an M word that happens to the DNA. Mutation, DNA mutation. What causes DNA mutations? You may as well say everything. I think everything causes mutations in cancer. Smoking causes cancer. Radiation causes cancer. Some chemicals cause cancer. But if any of these environmental agents damage your DNA, and if it damages it in an important gene, that could lead to cancer. So I look at it like maybe someone in the military. If you knew as a soldier you had to go attack the enemy, and there were 100 enemy waiting for you, What do you think your chances of survival if it was just you going after those 100 guys? Probably not very good, right? Because you're going to be aiming at one of them and all 100 are aiming at you. Chances of you getting shot are pretty high. But if you had 100 companions with you and there were only 10 of them, what are your chances now? It may be 1 in 10. Do you see it just a statistical thing? So by having this extra junk DNA scattered through your good genes, it's basically like a mutation sponge. The chances of a mutation randomly happening to the good stuff is reduced because you've just got this junk in between. But to make the gene, you got to get the junk out of the way because it's not going to be part of the code. So splicing happens in the nucleus to yield a mature messenger RNA that then is going to be shipped out that we build a protein with. So that was our messenger RNA, the code, the copy of the code. But we're making 2 other RNA in the process that we have to use to build a protein. Another type of RNA is called transfer RNA, and we abbreviate it tRNA. If we think of the messenger RNA as being the code that we're going to use to build and assemble our amino acids, and if we think of our ribosome, which is composed of ribosomal RNA, as the factory within which we're going to assemble the amino acids into proteins, we need the workers, right? The workers that are going to find the amino acids, bring them to the warehouse, and drop them into the assembly line in the right place. That's the transfer RNA. Each transfer RNA is going to be able to bind an amino acid, and think of it like it's carrying it on its head. And down where its feet would be on the transfer RNA, there are going to be 3 nucleotides that are going to match 3 nucleotides on our messenger RNA. And we call those 3 nucleotides on the transfer RNA the anti-codon. And this will all make a lot more sense once we start putting it together. But understand this transcription of DNA into RNA produces messenger, transfer, and ribosomal RNA. Each is going to be used in a different way to build a protein. Yes, ma'am. Yeah, here's a picture right here. So this is our model of transfer RNA. The top, this is the part where you bind an amino acid. So I'm thinking this is like the head. But down at the feet, there's this looping nature of our RNA molecule in which you have 3 nucleotides. Those are the anti-codon, and those are going to be used to line up with 3 nucleotides on our messenger RNA that are called the codon. We just haven't gotten to that part yet. So this is our transfer RNA. We've already seen our mature messenger RNA. And then here we have our ribosome, and inside the ribosome we're going to have 3 slots. We're going to have an A, a P, and an E, and we'll get to what those mean once we start building our protein. But you can see we're getting a little bit more complicated as we get ready to assemble our amino acids. So let's spend just a few minutes on codons and anticodons. Nucleotides are the genetic code. Our A's, T's, C's, and G's. Those alphabet letters are used to make up genetic words. And our genetic words consist of 3 letters. Each 3-letter combination of either A, T, C, or G is called a codon. And that codon is going to encode for either an amino acid, a start codon in which you're going to add the first amino acid of the sequence to start building the protein, or it's going to say stop. That's all the words mean. Start, add a specific amino acid, and how many different amino acids do we have? 20. Or the signal is going to be stop, in which case we finish building our protein. And when we look at all of the different words that we can put together out of our 4 nucleotides that are used to build our 3 words, that results in 64 different codons. Don't panic, you don't have to memorize this. You do not have to memorize this. I just want you to appreciate the diversity within the genetic code. So the way this is interpreted, our 3-letter word, our codon, you start in the middle of our wheel. See, that's 5', so that's your reading frame, that's your start. So if the first letter of our codon is A, you find the green A. Now, if we're also looking for the second letter, we go to the next level of our wheel. And in our genetic code, if the next letter is a T, what are we going to look for in our RNA code? A U. So here is an A, there's a U, and the third letter we're looking for is a G. So we look in this third wheel, our G is going to be red right here. That is going to be our start codon, which is also the codon for methionine. But we can do that for all 20 of our amino acids, as well as 3 different codons that signal to stop. So these 3 letters represent the code. That means on our transfer RNAs, those anticodons are going to have to be matching or complementary. So our anticodon for our start, if A is the code, what's going to be on our transfer RNA is the anticodon. A U, so that'll be a U. Here we have a U on the message. What's going to be on our A? Here we have a G. You're going to have a C. See, it's like matching the 2 strands of DNA. It's just for our RNA, those 3 words are going to be matched with the 3 nucleotides on our transfer RNA. So I want you to remember those 3 nucleotides are called codons. And I do want you to remember that AUG, that is the codon for methionine, which is the start codon to building a protein. So if AUG is the start codon for building proteins, what are all proteins going to start with? What's their first amino acid always going to be? Methionine. Now the cell may clip that out later before we get to a mature protein, but it starts with methionine. You have to have methionine for translation to begin. Now there are 3 codons that signal stop. The one I remember is tag. Tag, you remember freeze tag when you were a kid? Tag. You're done. Stop. Now that's the DNA code. In RNA, remember it's UAG, but I remember tag for stop. So I want you to remember AUG start and tag stop. Those are the only 2 codons I want you to remember. My bio majors have to remember all 64, but you only got to remember 2. Now, before we get to the actual stages and processes of translation, here is a static picture of how we begin. We've got our messenger RNA. See, there's our AUG. What codon is that? Start or methionine. It means the same thing. We have bound the methionine-containing transfer RNA, and there's the anticodon. So the U is matched with A is matched with U, and C is matched with G. Do you see the match? That's how the transfer RNA knows which position it goes in. Who's next? Once you bind that first transfer RNA to the start codon, the ribosome gets assembled around it. That's going to be the beginning stage. And that beginning stage puts that very first transfer RNA right in the middle, in the P slot. But here's the thing I want you to appreciate. Let me go back a slide. Do you see how we've got these amino acids like serine and glycine and alanine? It doesn't matter what the third nucleotide is. It's going to represent the same amino acid. So you don't have to have 64 different transfer RNAs for that to happen. That third level, you ever played Uno? You get that wild card? The third position for many of these transfer RNAs is like a wild card. It can match with anything. And that anything match is called the wobble effect. There's a special nucleotide position on our transfer RNA. You don't have to remember what it is. But it's a special one that has the ability to match with A, U, G, or C. And that's what enables that wobble effect. So alanine can be encoded by either a GCG, GCC, GCA, or GCU. It doesn't matter. As long as you get those first two right, that third one can be anything. And so that enables the same transfer RNA to be able to recognize multiple codons for certain amino acids. Not all of them. Some of them are pretty specific, like for methionine. It's only going to recognize AUG. But does that make sense about the anticodon? Does that help and see how it lines up just like the two strands of DNA would or the RNA copy would with the DNA? All right, now, translation is more complex than we saw with transcription. So here's another animation. And when I've shown this in the past first, it's really helped in the understanding of all the steps that we'll go through next.
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
All right, we got that? You got that visual? All right, so we basically have the 3 same steps we had for transcription. We see it again here in translation. Initiation, elongation, and termination. But the steps are different. Because for the initiation, we have to have the binding of that transfer RNA from methionine to the messenger RNA at the P site, right in the middle. That has to start it. Methionine is the only transfer RNA that's not coming into the A first. It comes into the P first. Once you bind the transfer RNA with the attached methionine to the P slot in the ribosome, the messenger's intact. Now the ribosome takes off. And it continues to allow new transfer RNA, which we've got our second amino acid coming into the A site. Then we have that methionine, the first amino acid, forms a peptide bond that gets transferred over to the one that just came in. Once that peptide bond is formed, now the ribosome slides. Now the second transfer RNA is moved into the P site. The empty methionine transfer RNA, because the methionine is on the end of the chain now that the second one brought in. That empty one gets ejected. The A site is open for the next transfer RNA to come in. And that keeps happening again and again as the ribosome walks along.
Does it become ribosome RNA when it meets the ribosome or after it comes through?
Ribosomal RNA. What the ribosome is ribosomal RNA. Just like the transfer RNA, it's RNA. It's a molecule of RNA. So the whole ribosome is ribosomal RNA. Now the messenger RNA, that's the code. That's going to be there first. The methionine transfer binds to it. Then the ribosome clamps around the whole thing, and it starts. Does that help? Okay. Now that keeps going and going and going, just like you saw in the video. New transfers coming in, the amino acids getting transferred to the growing chain, the empty ones getting ejected. That goes and goes and goes until the ribosome moves into a codon that is a stop codon. Because instead of a transfer RNA binding to a stop codon, there's a protein called release factor. I like to think of release factor like a stick of dynamite. Because when that release factor binds to a stop and that moves into the A slot of the ribosome, the whole thing blows up. The ribosome comes off, it releases the messenger RNA, and the protein is free. So that stops the process. When you get to that stop codon and you have that protein release factor present, that ends the process, the protein is done. And you've got other ribosomes reading other sections of the messenger RNA, making proteins behind it. It's like airplanes at Bush or DFW. Have you ever been at the airport and they've got those airplanes just lined up one after another? When this one lands, it better get off the runway because here's another one. Well, when this ribosome finishes, there's another one coming. It's got to get out of the way. And that process is going on over and over and over again. So, initiation in this illustration, we've got all of our components together. There's our methionine in the P slot. Here we've been transferring several amino acids. We have our newly arriving transfer. We're forming a peptide bond from the middle slot, and there an empty transfer RNA is getting ejected until we get to our release factor. You see how that looks very different than a transfer RNA. That's our stick of dynamite. As soon as that gets in place, this whole thing just disassembles, blows up, and our free protein goes off to do its thing, and we're just doing it again and again and again. Because we have multiple messages, multiple ribosomes, multiple transfer RNAs. Hang on just a second, got a question. So what happens to the empty transfer RNA? So if you've ever worked in retail, or if you've ever worked on an assembly line where you've got to bring stuff in, you got a whole bunch in the warehouse. You carry only one at a time. So the warehouse being the cytoplasm. Transfer RNA binds an amino acid and it comes in and says, okay, I need to put my methionine here. When it does and gets ejected, it's got to go get another methionine. So that cycle just continues to happen throughout the life cycle of a cell. So here's just a little cartoon showing different transfer RNAs attached to their single letter symbols for amino acids and showing our anticodons along with the codons. And I didn't want to make a big, huge, long one, so I just put our release factor here because remember TAG, AUG, that's our stop. That in a whirlwind is transcription and translation. Have a good weekend. We'll see you Monday.